I hope that I can reach you guys. Welcome back. I'm glad to see you guys. Beautiful day. I'm happy that you chose to come to class. Thank you for that. Today I will tell you about some of the sensory system associated with our ability to control our body. And the sensory system that I'm going to focus on is called proprioception. That's the sensory system that allows us to feel where our body is located in space. So if you've ever slept on your arm in a bad way so that you've cut off circulation to the nerve that goes to your elbow, what happens is that you wake up and your arm feels numb. What's part of that is that you can't feel where your arm is, and so you move your arm around a little bit, blood starts flowing to the nerves, and the nerves can once again send action potentials down from the sensors to the spinal cord, and then you regain your feelings about the body. So I'm going to tell you about proprioception today, and we'll do a little experiment to look at how proprioception gives you a sense of where your body is located. In this slide, what we have is a brief summary of the motor and the sensory system. So when you look at these extrafusal fibers, these were the muscle fibers that produce force when you activate them. They're controlled by alpha motor neurons, the motor neurons that sit in the ventral part of the spinal cord, send their axon via the nerve to the muscle fibers. They make contact on them via neuromuscular junction, and then what you get is one motor neuron connected to a bunch of muscle fibers, extrafusal muscle fibers. Then we have these intrafusal fibers that attach themselves in a parallel format to the extrafusal fibers, and they're innervated by sensory afferents called spindle afferents, and they signal length information to the spinal cord. The spindle afferents are wrapping themselves around these intrafusal fibers. Intrafusal fibers are muscle fibers as well, and they can contract as well, and they're controlled by these things called gamma motor neurons. And what we will see is that the gamma motor neurons, when they activate, they set the set point for the spindles, making it so that they measure length with respect to a bias. And that bias is described by the gamma motor neurons, where it sets the length of the intrafusal fiber. So when the intrafusal fiber stretches beyond that length, it signals that you have a length above the baseline. So I'll show you that in some detail. Okay, so we had alpha motor neurons that go to those extrafusal fibers. One motor neuron sends branches out to a number of muscle fibers, and so for your gastrocnemius, you may have like 10,000 muscle fibers that are controlled by one motor neuron. Well, it turns out that there are different kinds of muscle fiber and properties of the motor neurons as well. So you have certain muscles that are made up of almost entirely of what's called slow twitch muscle fibers, and then certain muscles that are made up of a composition of fast and slow. And these two kinds of muscle fibers have different metabolism, they have different fatigue properties, they have different innervation of blood flow and so forth, and they are going to define the function of that muscle. So there are two fundamental types of muscle fibers, one called type 1, which are slow contracting fibers. Here, these kinds of muscle fibers, if you repeatedly stimulate them, they produce a little bit of force, but they don't seem to fatigue, in the sense that the same stimulation repeatedly produces robust force regardless of how much stimulation is produced. Then there's type 2 fibers, which are fast contracting fibers. They produce force more rapidly with a higher magnitude, but they're fatigable type. So there's a fast fatigable and there's fast fatigue resistant. So type 2 is fast, type 1 is slow. The muscle and its function is going to tell you a lot about the composition of the fibers. So certain muscles, like our diaphragm, is made up of entirely of the slow twitch fibers. Never going to get tired of breathing. That makes sense, right? So you can't do a whole lot of force production with your diaphragm. It always does the same thing over and over, but it is always going to contract when you send a command to it. But certain other muscles, like your gastrocnemius, the muscles that you use for running, they will fatigue. And so after a while, you keep sending commands to them, and they're not going to move as much. And that is because they are made up of muscle fibers that produce a lot of force, but they can't sustain it. And so that's called fast fatigable. So eye muscles are another condition where they don't really fatigue. You don't get tired of moving your eyes. Okay? Extra musculoskeletal fibers, they are made up of different variety of fast and slow, and the percentage depends on the function of that muscle. So let me give you some examples of type 1 and type 2 muscle fibers. So here's a type 1 muscle fiber. So here's an action potential, this time here, and you see that it takes about 150 to 200 milliseconds for the force to reach its peak and then fall back down. This is a slow twitch fiber. Look at the magnitude of force it's producing, 1 gram of force. Now, if this muscle fiber is repeatedly stimulated, you see that it keeps on producing force, and gradually it builds up force. And now, if you keep on stimulating it for 60 minutes, you see that at the end of 60 minutes, it's still producing the same amount of force that it did at the beginning of the stimulation protocol. Here's a fast but fatigue resistant type of a muscle fiber. So here's the time of the action potential. You notice that it produces more force, about 10 gram, and it has a time scale that's about a little bit faster than what we see here with the slow type fiber. Here, you also see that it produces, after a period of stimulation, a measure of force. So here it's about 30 or 40 grams of force, but then after about half a second of stimulation, the force begins to decline. So you see that better here. After 6 minutes of stimulation, the force has declined by about a third, and after an hour, the force has declined by about 90%. Here's a fast, fatigable type of a motor unit, and here's the time of the stimulation. This is the action potential, and you see that it produces about 40 grams of force, so about 4 times the amount of the fast fatigue resistant, and its twitch is faster, so it rises up faster and falls faster, but it can't hold it. So you see that after a minute of stimulation, the amount of force that it produces falls to about 10% of its initial force. So this is fast fatigable, fast fatigue resistant, and slow. Type 1, type 2A, type 2X. So a motor neuron sends its axons out, and it makes neuromuscular connections to a bunch of muscle fibers. All of those muscle fibers and the motor neuron belong to a motor unit. That motor unit is going to be composed of either type 1 muscle fibers or type 2A or type 2X. So that motor neuron goes to the muscle fibers that all have the same properties: fast or slow, fast fatigable or fast fatigue resistant. Any questions so far? All right. Here are some properties of the motor units. So the slow type has axons that are small. So when an axon is small, what can you say about its speed of transmission of the action potential? If the axon is small, does it transmit an action potential fast or slow? Slow. The larger the size of the axon, the faster it will transmit the action potential. So the fast fatigable has a large size axon, and it will transmit the action potential more rapidly. Is that all right? Okay. The speed of contraction is slow in the slow type, fast in the fast type. The fatigability of the slow is little. It's resistant to fatigue. The fatigability of the type 2 is rapid. And the metabolism of the slow type muscle fibers is aerobic, whereas the fast fatigable is anaerobic. All right. So what does exercise do? So when you go to the gym and you lift weight, what does it do to this story of the muscle fibers? Well, you don't grow new muscle fibers when you go and you work out, what you're doing is you're enlarging the existing fibers that you have. So when you do strength training, you put stress on the tendons. This signals proteins to activate genes that make more myosin, resulting in enlargement of the muscle fiber. So you don't grow new motor neurons and you don't grow new muscle. The existing fibers begin to enlarge. Now, unless you have atrophy because you don't use it, then the muscle fibers will die. So for example, people in space, astronauts, when they go to space for even a short period of time, the lack of use of certain muscles because of the lack of gravity makes it so that there's atrophy. So there, in that case, an extreme version of it, you actually will get loss of muscle fiber. So you can think about the effect of extended training and the effect of lack of use as an extremes of this axis here, this x-axis here. So somebody who has a spinal cord injury would be on the left side of this axis, and somebody who's an endurance athlete would be on the right side of this axis. And so what this experiment has done is look at the quadriceps, which is a thigh muscle, and ask what's the composition of the fibers in the quadriceps when we look at somebody who is unable to move, basically. So you have somebody who is going to undergo severe loss of use versus somebody who uses that body part over an extended period of time. So what one observes is that people that have extreme endurance training, they tend to have a high composition of slow type muscle fibers in the muscle that they're using. So in the thigh muscle, you get this transformation to slow type fibers, the ones that are fatigue resistant. Whereas an individual who doesn't use that muscle at all, like an individual with spinal cord injury, you get a whole lot of fast type muscle fibers, the ones that will rapidly produce force but also not be able to sustain them. So the greater amount of use of the fiber tends to make that fibers transform into the fatigue resistant or even slow type fibers. Now, it's possible that people that are endurance athletes, they may be born with a preponderance of these muscle fibers to begin with. There may be genetic components that makes it so that they can transform the muscle fibers to the slow type fatigue resistant, or that the training itself may be sufficient. But probably there's a combination of the two. Yeah?
So in the scenario with the person with spinal injury,
yeah,
why do they only lose type 1? Why are those the only ones that atrophy?
So it's unclear why. It seems like, I don't know the answer to why. It appears that use is a trigger to transform muscle fibers to the ones that become less fatigable. Okay. It alters their metabolism. It makes it so that they become essentially more resistant to the action potentials that they get. Now, probably because of changes in the blood flow paths that are going, probably more vascularization is associated with it.
Okay. So are you saying it's like the default is type 2?
Yeah, the default is type 2. Yes.
More type 1?
Yes, in the quadriceps.
In your quadriceps.
Yeah.
Okay.
Now, probably not in the diaphragm.
Okay.
Yeah. Yeah.
So, like a person with a spinal injury, would they also lose like type 2X, just not nearly as many as type 1?
So the way this plot is made is that the sum of the three becomes 100. So this is proportion. It definitely is the case that there is going to be loss of muscle fibers. So muscle fibers are probably going to die as well. But what's left seems to be a greater proportion of type 2. There was another question back there. Same question, great. Any other questions? Okay, very good. When a motor neuron becomes activated, it sends axons to the muscle fibers, and then it engages those muscle fibers. Now, you have a large number of motor units that control a single muscle. You may have tens of thousands, millions of motor neurons that control your biceps. So when you go and you activate your biceps, how do these motor neurons become activated? Well, this plot shows you what happens as a function of voluntary force on the x-axis, and the y-axis is activity in each one of those motor units. So what you see is that when you're producing a small amount of force, you have one of the motor units become active at 8 Hertz, and then this motor unit increases its firing until a second motor unit becomes active. This increases its firing until a third motor unit becomes active, and so forth. So you see that voluntary activity is composed of two things. One, you have the activity of the single motor neuron increasing, and then you get recruitment of new motor neurons. So if you need to produce larger and larger force, first that single motor neuron becomes more activated, and then a second motor neuron becomes activated, then a third motor neuron becomes activated. So you get recruitment of these new motor neurons as you need to increase the amount of force you produce. So more motor neurons are recruited as you need more force, and the frequency of activation is increased. So what's the difference between the motor neurons that are recruited here versus the ones that are recruited here? To answer that question, there's a technique that's used that's called spike-triggered averaging, where what you're trying to do is figure out what are the properties of the motor units that are recruited early when you're producing a force versus late when you're increasing the amount of force you're producing. Question?
Can you go back to the previous?
Sure.
If you're looking at the first motor neuron and around like 20 grams it cuts off, does it plateau at that frequency, or does it just shut off at that?
Over here, this is probably, no, it doesn't shut off. It keeps on producing it, but that's probably as far as they were able to record from it.
Okay.
Yeah. Good question. Thank you. So I want to show you how can you tell what are the properties of these motor units as a function of time when they're recruited. Well, if you remember, when we talked about the slow and the fast type of motor units, we said that the slow has a twitch that takes a long time to develop, has a small magnitude. The fast ones have a fast twitch and have a larger amplitude. So if one could estimate the size of the twitch that's produced by a spike, then you would have a chance to say, well, what kind of a motor unit is this? Is this a slow type or is this a fast type? So how can you do that? Well, when you're recording from a combination of motor units, there's going to be a spike for every one of those times that a motor neuron fires. If you could somehow figure out what's the force that's generated by that single spike, you'd be able to tell the force response, this twitch response of that single cell. Now the problem is that there's going to be a whole bunch of spikes that are taking place. So here's the way to find the shape of the force twitch that's produced by a single motor unit. So suppose that we ask a subject to hold a force transducer between the thumb and their index finger. So they're going to be producing force with this, and we're going to be able to record it. And you put in electrodes, either surface electrodes or maybe a needle electrodes into the muscle that's going to be used to produce this force. So suppose that one could measure the spikes that are generated in the muscle as the force is being produced. So there's going to be a sequence of spikes. So here's one spike here, and here's another spike here, and so forth. Every time there's a spike, there's going to be a force that's associated with it. So presumably when I activate this muscle, when there's a spike, that motor unit is going to generate that little, action potential associated twitch. But the problem is that that twitch is going to be among a whole lot of other twitches that are adding up to give you the force. So what you're trying to do is to take out of your force measurements just the component that's due to that activity in this single motor unit. So you record these spikes, and then you also have the force. Then what you do is that you say, all right, let's take a window around the spike. So the spike is only telling me the activity in that single muscle fiber. So when you see a spike in that muscle fiber, look at the force around the time of that spike. Now, the force is the sum of all of the muscle fibers. So they're all activated. But the spike here is only from the single muscle fiber. So what you want to know is, okay, what did this muscle fiber contribute to the sum total? So if you were to do a triggering where you line up your data, every time there's a spike, you take the force and you line it up to the time where you got a spike, and then you average it over and over, you're going to get something that looks like this. So here's when you got the spike. Now you set your trigger. You say, OK, take the data around the spike. What data am I talking about? The force that you measure. And if you only had one spike, here's what the force looks like. If you had 10 spikes, here's what it looks like. 100 spikes, 500 spikes. You see, so after 500 spikes, you get a little twitch that corresponds to what happened when that spike came. It's called spike-triggered averaging. It's saying every time that there's a spike, line up your data to that time point, and then keep averaging it. Yeah, question?
So those are 500 spikes that you're laying over each other?
So there were, right. So you recorded, let's say, for 5 minutes from this muscle while the person was producing this force. So you have 5 minutes of force data, and then you have 5 minutes of spikes from this single muscle. Now, your problem is there are a whole lot of other muscle fibers that were also activated, right, when the person did this. You want to know what was the contribution of that single muscle fiber. So what the needle gives you is every time that there was a spike in that muscle fiber, you have a time now. You take your force data and you divide it up so that every time there was a spike, you have a window. Say, look at the size of this window. It's about 300 milliseconds. Okay, and so you do a triggering of your data based on your spikes. So you take your 5 minutes of data, divide it up so that every time you have a spike, you take 300 milliseconds of that data. And then you...
The spike from the EMG is the needle.
Yes.
One. So
yes.
It's in the muscle fiber.
It's in the muscle fiber.
And then the tension, the force is...
Between the two fingers.
Well,
yeah.
Like the whole muscle.
That's right, the whole muscle.
So you're just trying to find, so by lining it up, you're just finding the spots that you're looking for.
Right. So the analogy that I make is as follows. Suppose that you guys are all talking at the same time. Suppose that you were. But I'm interested in hearing one person. It would be impossible for me to do it, right? Because you're all talking at the same time. So what I would do is that any time that that person is talking, I would ask them to press a button. I would take the audio recording that I've made, and every time they press that button, and let's hope that they say the same thing over and over. Hello, hello, hello. I would be able to pick out their voice out of that whole total because I would know how to take out the noise, which is everybody else's voice. I would need to know the timing when they spoke and then average every time that they spoke. And that would give me just their voice. Everybody else would be canceled out. Does that make sense? That's what this technique is. Okay, what's the purpose of this technique? What we wanted to know is that what's the characteristic of the twitch response of the motor units. So now we have this technique called spike-triggered averaging. Now, suppose that you record now from muscles and you see that here's an action potential and then here's another action potential that comes. So this is from one motor unit, here's from another motor unit. And what you want to know is that what's the twitch associated with this unit and what's the twitch associated with this unit. So what you do is just, again, you do spike-triggered averaging. So every time you have a spike from this unit, you take your force data. Every time you have a spike from this unit, you take the force data. And after you average, so here's the force that's generated by the whole muscle. You see that the force is increasing. This unit becomes active when the force is large. This unit becomes active when the force is small. And if you were to compute the force, the spike-triggered average response of the muscle, you would see that the first unit has a twitch that is a small magnitude, long duration. Second unit has a twitch that has a large magnitude, short duration. So what one sees is that you're getting a fast response from a unit that was recruited later and a slow response from a unit that was recruited earlier. So probably this is a slow twitch unit, this is a fast twitch unit. So the natural progression of recruitment is to go from slow, fatigue-resistant motor units, and if you need more force, you eventually recruit the large, fast force-producing, fatigable type of motor units. Questions? All right. So there's a principle here which tells us something about how you can identify characteristics of neurons in the motor units in the motor system. By spike-triggered averaging, you get an opportunity to determine basically the properties of the forces that they produce. In another way, you can determine when you're recording from the brain, say that you put an electrode in the brain and you record voltages and you see spikes. Well, it turns out that if the neuron is close to your electrode, it's going to give you a shape for its spike, and if the unit is far from your electrode, it's going to give you a different shape associated with the spike. So that's how you can tell that if you get a spike that looks like this, it came from this unit. If you got a spike that looks like this, it came from someplace else. And basically, spikes that look the same probably came from the same neuron, and spikes that look different from the one you got comes from a different neuron, because their distance from your electrode is going to affect the shape of the voltage that you get. And that's how you can tell that you're recording from two units. This is what you see here. So here's a spike from unit 1, here's a spike from unit 2, and they have a different shape. In this one's negative, this one's positive. And the shape comes not because the action potentials are different, right? The action potentials are the same. It's because the distance of the unit to the electrode is different, and because of the impedance, you get the different shape. Any questions? All right. So almost all of us are handed individuals. About 90% of us are right-handed. So one can ask, is there a difference in the motor units that we have on our preferred hand as compared to our less preferred hand? So we saw that in an individual that was an endurance athlete, you had a greater proportion of fatigue-resistant type of motor units. So you use your preferred hand more than your non-preferred hand. Does the composition of the muscle fibers in your preferred hand differ from your non-preferred hand? And the answer is yes. It turns out that if you look at the recruitment threshold of the motor units in your preferred hand, it tends to be lower than the recruitment threshold for your non-preferred hand. It's easier to recruit those units, which would suggest that the composition of the units have changed because of the way you've used it. Then there's probably greater proportion of fatigue-resistant type motor units in the hand that you prefer to use. Yeah?
Now, when you say the hand, are you looking at the motor units in the entire hand, or are you looking at specific muscles?
Yeah, this experiment was done on a specific muscle, and to be honest with you, I don't remember. I think it was probably flexor pollicis radialis, the muscle fiber that was being used. And you'd have to look at the specific paper to see which one they used. Yeah, I think it's FPR, but I'm not sure. Just one muscle. Dorsal interosseous muscle, sorry. Yes, there it is. Yeah. Okay. All right. Let me tell you about the sensory system of the muscles. So here's the extrafusal fibers, these large muscle fibers that attach to the tendon. Here's the tendon region. And here's an intrafusal fiber, as you see here. And these are the muscle spindles that wrap themselves around the intrafusal fiber, send the sensory information about the length of the intrafusal fiber back to the spinal cord. This is called muscle spindles. The muscle spindle afferents tell us information about the length of the muscle fiber. In the region that connects the extrafusal fiber to the tendon, there are another kind of sensors called tendon organ afferents, Golgi tendon organ afferents. And it turns out that these afferents here that innervate the extrafusal fiber's junction with the tendon, they measure force that's being produced by the extrafusal fibers, whereas the muscle spindles measure length that's produced by the extrafusal fibers. So muscle spindles signal length to your spinal cord. Golgi tendon organ afferents signal force that's being produced by your muscles. I'll show you these two things in some detail. So here's a muscle spindle afferent. And we're going to look at its response as a function of change in length. So here's the length of the muscle. It's being stretched and held constant. And what you see is that the muscle spindle increases its firing and then drops its firing during this period of constant length. So it's particularly sensitive to change in length, as you see here. Its discharge is higher here than here. It is particularly sensitive to this period where the length change is taking place. So this is muscle spindle afference, and there's a change in the length. It's signaling that there's a change in the length of the muscle. Here's a Golgi tendon afferent during a period where there's a change in force. So this is what's called an isometric condition. So the arm is kept isometric, it's not moving. The individual is increasing the force by activating the muscle activity. And what you see is that as the force increases and maintained, the Golgi tendon organ afferents increase their activity, maintain it, then they drop off. Golgi tendon organs, those neurons that are encoding force, send the information to the spinal cord via the dorsal root ganglion. Spindles encode length and also send their information to the dorsal root ganglion to bring the information to the spinal cord. So here's an experiment that you can try yourself. Your cell phones have a vibrator mode, and what vibration does is that it activates muscle spindles. Your muscle spindles are particularly sensitive to vibration. And in this experiment, what's going to happen is that you're going to be asked to basically try to tell the experimenter, when you close your eyes, where's the location of your arm. So if I close my eyes and you place my arm here, I can pretty well match it with the other arm. You know, I can do a very good job of matching it. So if you now place a vibrator, like your phone, on top of your belly of your muscle, what you will do is that you will activate those muscle spindles. They will increase their activity when they feel the vibration. What that tells your brain is that the length of this muscle is longer than it really is. So if you were to ask me, when you are vibrating my muscle spindle, to tell you where is my elbow, I would do this. Because I feel as if my muscle biceps is longer than what it actually is. And the illusion is really strong, because if you were to hold your nose like this while you are stimulating your biceps, it gives the illusion that your nose is growing. Pretty cool. You have to try it. But you have to get the muscle spindle activated. Okay, any questions? So your sense of where your body is comes from the activity of these muscle spindles. Okay, now, like most sensors in our body, our nervous system controls the sensitivity of the sensors. So muscle spindles don't measure the absolute length of your muscles. They measure the relative length with respect to some baseline. And that baseline is the length of the intrafusal fiber. So remember, muscle spindles wrap themselves around this intrafusal fiber. As the fiber gets stretched, the spindles fire. They're sensitive to the stretch. Well, the length of those intrafusal fibers is controlled by a motor neuron in your spinal cord. It's called the gamma motor neuron. Basically, the nervous system sets the sensitivity of those muscle spindles by activating the gamma motor neurons. When the gamma motor neuron activates, it shortens the intrafusal fiber. Now, what happens is that if the extrafusal fiber are lengthening, it of course also lengthens the intrafusal fiber, and the muscle spindles are vigorously activated. If, on the other hand, the gamma motor neurons are not activated, then what happens is that the intrafusal fiber goes slack. So now you need a much larger change in length to be able to sense the activity that has taken place. So by manipulating the length of the intrafusal fiber, is it short or is it long, you set the bias above which you can sense length changes in your body. And it turns out that when you make movements, your nervous system coordinates activity of these gamma motor neurons with the alpha motor neurons. Alpha motor neurons produce the force. The gamma motor neurons are telling you how sensitive you are to length changes, how well you want to be able to respond to it. So in this slide, what we have is that a little cartoon of this scenario. So here's the spinal cord. Remember that butterfly shape region? That's where the motor neurons are. So here's a motor neuron here, the alpha motor neuron. It's going to go to the extrafusal fiber. And here's a gamma motor neuron. It goes to this intrafusal fiber. It regulates the sensitivity of the muscle spindle. The muscle spindle wraps itself around the intrafusal fiber. It's sensitive to length change. It brings the information back to the spinal cord. So here's the cell body. It's sitting in the dorsal root ganglion. Brings the information back in the spinal cord and acts on the alpha motor neuron. This is an excitatory synapse here. So what this means is that when there's length change here, so when the length of this muscle becomes long, you get activity in the spindle. That spindle activity goes and excites the motor neuron excites the muscle contracts, it resists the change in length. So when something pulls on your arm, your alpha motor neurons naturally respond to that pull by contracting. Why? Because when something pulls on your arm, the muscle gets long. This muscle is going to get long. The spindles are going to sense that. They're going to send the information back to the spinal cord and via a single synapse act on your motor neurons, and that causes contraction of that muscle. This is called the stretch reflex. It's the most primitive form of a feedback control that you have in your body. Your single fastest way to respond to a sensory stimulus is this pathway. When something pulls on your muscle, it activates the spindle activates the motor neuron contracts the muscle. It's called the stretch reflex. It's a simple control system. Okay. Now, what about those Golgi tendon organs? So remember, we also have another sensory system that measures force. The Golgi tendon organs have their process here in the tendon area where the extrafusal muscles fibers connect to the tendon. They sense force. So when there is force being generated, this system becomes activated. The tendon organ becomes activated. It activates an inhibitory inner neuron in the spinal cord, which then inhibits the motor neuron. So basically, this is another feedback loop. As force increases in the muscle, this shuts down the motor neuron. It reduces its activity. Okay, so let me show you that in the summary slide. So you have two pathways for feedback control. When you generate force on your muscle by activating your alpha motor neurons, you have the tendon organs that measure force. They send this force back to an inhibitory inner neuron that inhibits that input. When that force acts on your arm, it changes the length of the muscle. That length is sensed by muscle spindles, which are regulated by gamma motor neurons, which then excite the alpha motor neurons. So you get length and velocity feedback via the spindles. You get force feedback via the tendon organs. Any questions? Okay, so suppose you go to your general practitioner and they are testing to make sure you're healthy. One of the things they always do is to have you sit down and they will hit you right around, let's see if I can do it. There we go. They check your stretch reflex by hitting your tendon below the patella. So what are they doing? What they're doing is that by hitting this tendon, they cause a stretch of the muscle. That stretch is sensed by your spindle afferents. The spindle afferents increase their discharge because the intrafusal fiber got stretched. They send that activity via the dorsal root ganglion cells to the spinal cord, and after a single synapse, they activate the motor neurons that goes to that same muscle, causing it to contract. So there it causes this muscle to contract, it lifts the leg up. Any questions? Good. Okay. So the spinal cord has this feedback mechanism. Force feedback via the Golgi tendon organs, position feedback via the sensors from the muscle spindles. Well, that information also reaches your brain. And what I want to show you are some information about the time delay in these cases. So in this experiment, what's happening is that the task is to basically pay attention to the instructions on the screen and either oppose the perturbation that's going to come or assist the perturbation. So your biceps is suddenly stretched at some time. So you don't know what time they're going to stretch the biceps. They're going to pull on your arm. And what you have to do is pay attention to the screen. The screen tells you to either resist this perturbation, push back, or assist the perturbation. Let it go. Okay? So let's see what happens. So this is the EMG from the biceps. What you see is that here's the time of the stretch, S here, and about 50 to 100 milliseconds after the stretch, there's this response in the muscle. And that's there regardless of whether the instructions were to oppose or to assist. So there's a component to this reflex that's outside your voluntary control. You're going to respond to it anyway. After that, there's another component that you see here. So this is the voluntary response. And this depends on your ability to respond based on the instruction. So this secondary response is because of the signal that's going to your brain and coming back down to your spinal cord. This first response is just to the activity that's taking place in your spinal cord. And you can see that the delay for the voluntary response is quite long. It's 150 milliseconds or more. All right, so there's a short latency pathway where sensory information comes in the spinal cord, acts on the motor neuron, goes out to the muscle, and there's a long latency pathway where that sensory information gets a copy of it sent to the thalamus, to your motor cortex, and then via the cortical spinal tract comes back down to the motor neuron and acts to the muscle. So this is the fast response pathway. This is the slow response pathway. And the slow response makes it so that we can't voluntarily respond to stimuli nearly fast enough, which is, what the problem was when somebody lifts this off my arm. When they lift it off my arm, I can't hold my hand nearly as still as when I lift it off my arm. So the reason why I can do it is because when I am moving this thing off my arm, my brain is predicting precisely when that's going to happen, and it's deactivating my biceps at precisely the right time. It's rather striking, I think, how well the system is calibrated and how sensitive it is to small changes. I mean, just to illustrate to yourself how sensitive your body is to small miscalibrations, is all you got to do is, pick up at something that you expect to be heavy, but it's light. And only a pound difference in the weight of something you will see that you make an error in your movement. We're very exquisitely fine-tuned. Okay, any questions so far? All right. So one can measure some of these time delays, and here's one way to do it. So in this experiment, the ankle of a subject is suddenly stretched. And the reason why the ankle is shown here is because that has the longest delay, because it's the farthest away from my brain. The time it takes to activate things in the ankle is going to be longer than it takes to get to the fingers, for example. So the ankle is stretched, and here's the stretch of the ankle. You see it down here. And then here's the EMG that's resulting here. There's some EMG that occurs early, some EMG that occurs late. And we can divide up these responses into a couple of components. So if you were to stimulate the brain, and you record how long it takes for it to generate an activity in the muscle, this is shown here. So you can put a transcranial magnetic stimulator over the medial part of the cortex, so you activate cortical neurons that send their projections to the spinal cord that eventually reach the lumbar part of the cord, and that then goes to the ankle muscle. And you see that it takes about 30 milliseconds after the stimulation. So here's when the stimulation takes place, and here's when the response is shown in the muscle. So the delay from here, activity in the neuron, to the muscle is about 30 milliseconds. On the other hand, if you were to move the ankle, and then you record how long does it take to see activity in the cortex, so the ankle is moved, and then you record via EEG how long it takes to see activity, it's about 50 milliseconds, which is shown here. So this is how long the sensory system takes to provide information to the cortex. This is how long the motor system takes to go from the cortex to the muscle, which means something like 80 milliseconds or so of this stimulus time is taken up from the activity of just the spinal cord and the brainstem. And here's the part where it's probably going through the cortex. So these first two peaks here probably are due to local reflex pathways in the spinal cord and the brainstem. This is probably something that's coming from the brain, the cortex. So there are delays in these sensory systems and the motor system, requiring the system to predict. And based on that prediction is the reason why you move so accurately. Any questions so far? Yeah, no questions? Yeah, yes, sir.
Is there any advantage to these delays?
They are basically a consequence of evolution. We cannot choose the way we are made, and so today robots are, of course, are made without such delays, right? So if effectively you are limited by the time it takes for these action potentials to go through the axons, which is basically the speed of sound. Whereas, when you make machines, wires carry your electrons at the speed of light. So it's particularly astonishing how well we move compared to robots, given that we have such slow transmission lines. And the reason why we move so well isn't because we have great wiring, it's because we have an incredible brain. It has learned how to control your body. So one of the lessons of evolution is that, you can only change slightly what you're born with. You can't redesign. And the animals that describe our history, they started out of having no nervous system at all, and the nervous system formed, and none of them really walked upright, and all the problems that we face today. And yet we have this magnificent thing that that allows us to function in the world. All right, just a couple of more things before I'll let you go. So I mentioned to you that there's a disease that can affect the sensory system, and this is the proprioceptive system. It's called large fiber neuropathy. Large fiber neuropathy causes death of the muscle spindles and the neurons that bring afferent information, proprioception, to the body. What I want to show you is that these individuals, they can make movements, but what happens is that their movements fall apart when they don't have visual feedback. So in this experiment, what happens is that a healthy person is asked to make a thumb flexion. So just a flexion of the thumb. And here's the position of the thumb, here's the velocity of the thumb. So here's the movement that takes about 150, 200 milliseconds, and you see that it has this agonist burst, antagonist, back to the agonist. That 3-burst pattern that we had seen in last lecture. Here's a person with large fiber neuropathy. It's called deafferented, meaning that they've lost their afferentation. They can also make a fast movement, and you see here's their velocity, and here's their agonist, antagonist, and a little bit of agonist again. So that activity that we had seen in healthy individuals can also be present in a patient that has lost sensory feedback. So this pattern here is not coming about because you have sensory feedback during the movement. It's because your brain is making predictions about how to make this movement and is programming these bursts of activity in a feed-forward way. Now, what happens to the movements of these individuals when you remove the visual feedback. So to demonstrate this to you, here's an experiment that shows it. So if I were to ask you to produce a position, so you move your thumb and hold it in that location. So if you were to produce such a position and now we would turn off the lights so that you couldn't see your thumb anymore, you could still hold that position. You know, so you could do this, you could close your eyes, and you could still maintain that position. However, in people that have lost proprioception, they can make that movement, but you see that after the vision is removed, their hand drifts. So without visual feedback, they can't hold that position. All right. Are there any questions? Very good. See you guys on Friday.